Thank you so much for having me. It's an absolute privilege to be here for the Open Science Day, and I'm so sorry that I can't be in there in person in order to share with everybody about one of my absolute favorite things in the world, which is RNA modifications. Cells face the daunting task of having to maintain the right number of proteins in the right place at the right time. And one general strategy they use to do this is to modify or chemically tag all three major classes of biomolecules in the central dogma in order to control their structure, function, and stability. And we tend to talk and think a lot about the sorts of modifications that get put into DNA and proteins, things like methylations or phosphorylations. But as we've heard about during this session, it turns out that RNA, that central molecule in the central dogma, can also be highly modified. And there's actually been over 150 different varieties of RNA modifications that have been discovered. And we've known about the existence and the importance of modifications in RNAs since the dawn of RNA research in the 1950s, when some of the most prevalent modifications found across biology were first discovered. And since the 50s, the dogma in the field, until quite recently, has been that RNAs are highly modified. These modifications are important, but that they're mostly just the purview of non-coding RNAs. And just to remind everyone in the audience, a non-coding RNA is just a catch-all term for every RNA in the cell that is not a protein-coding mRNA acting as a blueprint for protein synthesis. And these non-coding RNAs have been easier to look at their modification status simply because they are much more prevalent than mRNAs, with non-coding RNAs comprising 98 to 99% of the RNAs by total mass in a cell. However, thanks to technology, in particular deep sequencing technology, the last 10 years or so has really flipped this long-standing dogma on its head, as scientists who are interested in modifications have taken advantage of transcriptome-wide sequencing techniques in order to develop methods for mapping their particular RNA modification of interest across all of the RNAs in the cell, not only the abundant non-coding RNAs. And as they did this, they've discovered that protein coding mRNAs, particularly in eukaryotes, are highly modified. And to date, far more than 10,000 sites of modification have been identified in eukaryotic mRNAs. And this discovery has really breathed new life into the RNA modification field and ignited the imagination of scientists worldwide, because these small chemical modifications have the potential to change every step in the life cycle of an RNA after it's been made in order to control their structure and dynamics, their splicing and maturation, their final form, their half-life or stability in a cell, how they interact with other components of the cellular machinery, like other RNAs or proteins, in order to ultimately control or influence the function of a given RNA molecule, and perhaps provide a really essential hidden mechanism of gene regulation. And in line with the idea that these modifications might be playing an important role, enzymes that are responsible for incorporating modifications into both non-coding RNAs and mRNAs, their dysfunction is linked to a whole host of diseases ranging from neurological disorders to cancer, and also to a wide variety of essential cellular processes such as development, circadian rhythm maintenance, protein homeostasis, and immune response. So these modifications and their enzymes that are incorporating them are clearly very important for biology. And the realization that they are so important and so fundamental has recently been recognized in a study that was put out last year from the National Academies of Science. I have a QR code linking to that study here. And in this study, the NAS proposes that we need to really be investing in new tools to study RNA modifications, because they really present a brand new avenue for the development of new therapeutic targets and new therapeutic technologies. However, in order to realize this future, we need to first be able to figure out where modifications are in biology and assign specific roles to those modifications. I want to point out before we begin that even though we know there are something like way more than 10,000 sites of mRNA modification, we really don't know what any of these modifications do functionally. Even for non-coding RNAs that have been studied for 50 years, we have only barely scratched the surface of what these modifications are doing. So the questions that we have to ask ourselves as we're trying to reach this new future, this new goal of being able to take advantage of our knowledge of RNA modifications, is to be able to ask a couple of fundamental questions. And today what I want to talk about are what the tools are that are available for answering these questions and what tools it is we really need in the future going forward in order to tackle them. So the first thing that we need to know is what I think is something that should be quite obvious, and that is, what is there? We cannot study anything that we don't know that exists. And there are still new RNA modifications being discovered every year. So we clearly don't have a wide-ranging picture of the entirety of RNA modifications that are possible in a cell. We also need to know not only what's there, but where it is and how it got put there, because knowing something exists and knowing where it is are both really important pieces of information when you're trying to identify what's the biological role of a particular chemical modification might be. We also need to know all of this information in a quantitative way, because if you're going to pick, your favorite point modification out of 10,000 different sites and try to decide which one might be most biologically consequential, it would be useful to know if that modification is in every single RNA of a particular sequence or if it's only there 2% of the time. Lastly, all of this information needs to come together with fantastic biology and biochemistry and be pulled together in order to figure out what the function of individual sites are, which I think should really be the ultimate goal of the technologies it is we're all striving to develop. So I want to start off by talking about the most robust and traditional way of measuring and identifying RNA modifications. And that is through global ribonucleotide modification. Profiling, or GRMP. The techniques that have been used in order to do this can really give us information about what is there and how much of it is there. These tend to be more quantitative than the next generation sequencing technologies. And all of these approaches share that they begin with an RNA of interest, which has either been isolated individually or comes from a pool of RNAs. And these RNAs are then treated with enzymes that chew them down into their building blocks, into nucleosides. There's then some sort of a targeted quantification and analysis in order to look for which modifications are present. So all GRMP methods start the same way, where they chop down an RNA or pool of RNAs of interest into their molecular building blocks. This is done using enzymes, nucleases, and often also alkaline phosphatases. The most long-standing way of doing this is to then take your pool of ribonucleotides and subject them to TLC, thin layer chromatography, which some of you may remember from way back when you took organic chemistry. And in order to run these assays, they're done in 2 dimensions, where the ribonucleotides in the samples separate on a TLC plate in 1 dimension. That TLC plate is then turned into a second solvent, and they're separated again in a 2nd dimension. This has been done since the 50s and yields things that look like this. So shown here is some actual data from a paper where they were looking to try to figure out what modifications or what enzymes are putting a particular modification into E. coli rRNA. And you can see each one of these spots here is corresponding to a different nucleotide, modified or unmodified. And this spot here the M2A spot goes away in RNAs that have been purified from this delta YFGB strain. So this is a very powerful technique. You can see right away that that spot goes away. That must be what it is. But you can imagine how laborious it would be to go through an organism and knock out every possible enzyme and look to see which spot disappears. Luckily, a new technology has come online since then called liquid chromatography coupled to tandem mass spectrometry, or LC-MS-MS. And LC-MS-MS uses the same principal ideas, where we start off with a pool of digested ribonucleotides, only now, instead of separating them by 2D TLC, we separate them by a more modern technique called liquid chromatography, or HPLC, which I'm certain many in this audience are likely familiar with. This technique was really pioneered and widely implemented by Jim McCloskey, Pat Limbach, Mark Helm, and Peter Dedon's lab, as well as many, many others. This now is pretty much the standard for doing modified nucleoside analysis in RNAs. So once you've separated out your pool of digested ribonucleotides, you then take it and you inject it into a mass spectrometer, and you look at each peak in your LC chromatogram, and you can see the different masses for that peak, and particularly exactly where they're coming off. And this can allow you to identify, in particular, sites of modification. However, you have to know what sites you're looking at before you begin. You have to know what the modification is. We have to do LC on the front end because the level of modifications is so much lower than the level of canonical nucleotides. When I talk about this, I always like to show this LC trace from my lab, where you see we've taken total RNA from yeast and we've digested it down into nucleotides. And you see large peaks for the canonical 4 bases, and you see these tiny little peaks underneath it. Those tiny little peaks are all of the modification. So if you were to take your mixture and just put it directly onto a mass spec, you would not be able to see signals for all of these less abundant modified nucleotides because you would have ion suppression. So doing the LC on the front end is extremely important. We can also look for new modifications by LC-MS-MS, but this is much more challenging. Here in this particular scheme, if we know where a modification elutes and we can run a control sample against that known modification, we can very rigorously tell what modification is there and how much of it there is. However, if we're looking for something brand new, we have to take a much more sophisticated approach. In this approach, we do an unbiased type of LC-MS-MS, where again, we're starting with nucleotides. We are then taking 2 MS spectra. So you put it into the orbit trap and you use the MS1 to get a chemical formula prediction. You then pick ions in that MS1 and do an MS2, and you do CID or HCD, which is a collision sort of follow-up in order to break the molecule apart further and assess the masses. And this can give you an idea upon ribose loss, what exactly is modified. Is it the base that's modified? Is it the ribose that's modified? And in this high accuracy mass spectrometer, you can tell what the exact mass is. An example for what these different fragmentation patterns look like are shown here with M6A and M1A. And I think it's important to point out that this HCD or CID is extremely important because these 2 modifications have the exact same mass. Both have a methyl added to an adenosine, just at different positions, but they fragment differently because of their positions. If we don't know what a modification is going in and we want to see where things are connected to each other, we can do this entire process using labeled nucleosides that we collect by growing our cells on labeled media. And this can allow us to identify the ratios of carbons to nitrogens in a modification, and whether or not that ratio changes in the base or in the ribose. So this is a very powerful but extremely laborious process for discovering new RNA modifications. So together, these global modification profiling strategies are fantastic in that they are direct. They're directly sequencing RNAs. They have high throughput. They're highly sensitive. They're quantitative, and they have all sorts of internal sample controls. But they unfortunately provide no sequence information and are incredibly, or can be incredibly laborious. They also take a huge amount of material relative to sequencing studies. So the next question we want to be able to ask is, where are modifications and how are they added? In order to do this, we need sequence information that isn't accessible to us in those sorts of global ribonucleotide profiling assays. So really the first leaps into this area were done using DNA sequencing technologies, in which some sort of reverse transcriptase was used to convert RNA into DNA, and how it converted that RNA into DNA changed based on the identity of the modification and allowed us to see whether or not a modification of interest was present or missing from a particular site. So for example, there's direct detection where there's a modification that changes the base pairing face of a particular nucleotide that leads to the misincorporation of a nucleotide. And when you do the sequencing and you do the alignments, you can see a change in the base pairing from the expected base. There's also chemical assisted where you chemically treat the RNA in order to change base pairing or in order to call an RT stop, which can then be looked at in a sequencing. You can also use an antibody in techniques like RIP-seq and CLIP-seq in order to cause the either the RT to stop or to pull down pools of RNAs that contain a particular modification of interest. And this is probably the most widely implemented sort of technique that's used for looking at where modifications are within an RNA sequence. The pros of these sorts of techniques are that they are transcriptome-wide, they're sensitive, and very little material is needed. And I just want to highlight that these techniques have really laid an important foundation for the RNA modification field, particularly in mRNAs and for the transcriptome-wide mapping of RNA modifications that we're all now continuing to build on. However, these techniques are not perfect. Their main cons are that there's been problems with reproducibility between methods. The number of modifications detectable per method is very limited. The readout is indirect. You're reading DNA, not the actual RNA it is you're trying to analyze. These methods are rarely quantitative, and they all have short reads. So if you see a signal, you can't get an idea of whether or not there are multiple modifications along an RNA. All you can tell is that that particular spot can be modified. A way to get at this more directly would be to use LC-MS technologies, which are similar in many ways. In this case, you take a pool of RNAs and you use enzymes that only digest those RNAs into particular fragments. This is much like bottoms-up sequencing for proteomics. You then separate them out using an LC and use high-resolution MS or mass spectrometry in order to sequence each one of those fragments. The pros of this is that it is directly analyzing RNAs, it's sensitive, and it's a gold standard. The con is that it takes huge amounts of material. It can only look at simple mixtures or individual molecules. The analysis tools are quite poor. The RNases are not as precise as proteases in proteomics. You don't get full coverage, and you're again only looking at short reads. So I'm not going to spend a lot of time talking about what the latest and greatest sort of future forward thing is in RNA modifications, because it's been talked about so much here, and that is nanopore sequencing technology. Nanopore technology for detecting modifications in its current form has largely relied on looking for errors in base calls and looking and seeing that spots that have high error rates may have putative modified sites. More recently, the future and sort of the bleeding edge of that field is to move away from looking at errors in base calling, instead to looking at the actual raw signal intensities for particular sites and comparing them with a non-modified control that's experimentally generated in order to identify the sites of modification. Nanopore technology is great because it's transcriptome-wide. It's directly reading RNA. It gives you long read information across an entire molecule, and it takes relatively low material to input. The biggest cons are that there's biased information on what's there. You can only find what you're looking for. It's not quantitative. And right now, it still has a high false positive, false negative rate for calling RNA modifications. So I would say my summary is that no one technology can do it all, and that the rigorous de novo discovery and quantification of RNA modifications still really remains very difficult, and that we're going to need to integrate all of these platforms in order to generate the information we're going to need in order to ultimately develop new algorithms for being able to access the information we're collecting in nanopore sequencing and bottom-up LC-MS sequencing methodologies. So in short, the future of RNA modifications is really to integrate GMRP modification sequencing with site-specific stoichiometry and biochemical analysis in order to understand the molecular level consequences of RNA modifications and meet that vision that was recently laid out for us in that NAS report. And I'd like to thank my lab and all of you for your time.
Thank you so much, Kristin. Do we have questions? One I have is, not all modifications are equally abundant, certainly. And we also don't know as much about all of them. But of those that occur in mRNA, is it make sense to try to focus work on a handful for which there's the greatest amount of biological information and insight? Or do you think it's really more important to, if you have to choose in a resource-constrained world, go broad or go deep?
I think deep. I think deep for sure. Although I just want to point out that when you say the ones we know the most biological information for, that's really only M6A.
I missed the last, but can you say that again?
Well, I would just like to say, when you say the ones we know the most biological information for, I would point out that that's probably right now just M6A.
M6A, okay.
And maybe an inosine, right? But anything else, we know it's there, but there haven't been the tools yet to show what the biological role of those things precisely are. So I think my approach would be not only to go for the ones for which we have biological information, because we're certainly biased by looking at things we already know how to see, but by instead looking at the ones that are most abundant. I think I would take it in order of abundance if it was me.
All right. Gene has a question.
Kristin, this is Genio. Yeah, so great talk. I think one of the things that's missing is our tools that can allow site-specific erasing and writing, right? Because then you can see in the cellular context whether that specific mod is important for whatever process, right? And I think that's a piece that hasn't seen a lot of light, even though there are some technologies that are starting to evolve. I wanted to see if you could comment on that. Yeah.
So I think that's a smart idea for methylations. I think it's going to be harder for other abundant modifications like isomerizations, which aren't going to be erasable. For example, I can't see pseudo U ever being erased. The way that the field gets around this right now is to use knockouts, but of course that's like hitting something with a sledgehammer.
Yeah,
I totally agree with you that's great, but I think we're a long way away from there.